Okay, so we will now transition to our first presentation of overview of the FDA medical queries. Next slide, please. So as mentioned earlier, I'm Vaishali Popat. I'm the Associate Director for Biomedical Informatics and Regulatory Review Science in Office of New Drugs. Next slide, please. So joining today with me as presenters are Dr. Scott Prostel, who is a Senior Medical Officer in the Biomedical Informatics and Regulatory Review Science team in Office of New Drugs, and Dr. Eric Brodsky, who is the Associate Director of Labeling Policy team in the Office of New Drugs. Next slide, please. So I will provide introduction and background, and then Dr. Prostel will talk about algorithmic FMQs, and Dr. Brodsky will talk about labeling of grouped terms. Next slide, please. So let's look at why FDA medical queries. So imagine a patient in the clinical trial develops abdominal pain and goes to the one site, and there the investigator physician writes down abdominal pain in the case report form. And that is actually considered, whatever the investigator writes down is considered a verbatim term. Then that verbatim term gets coded using standardized MEDRA referred terms. So then let's imagine the same patient goes to a second site, and this investigator writes down upper abdominal pain, and the third investigator writes down lower abdominal pain, and let's say the fourth investigator writes down abdominal discomfort. None of this is wrong, medically, but they're all similar clinical events. So all of this, because MEDRA has a lot of preferred terms, are more than 24,000, so it's very granular. So there is a preferred term for each one of this, so they would be coded into different preferred terms. Now, adverse events also may manifest in related but different ways. So a patient with rash related to drugs and hypersensitivity may present with an erythematous rash, or a macular rash, or a maculopapular rash, or a papular rash. And each one of these would also be coded to a different preferred term. So when related preferred terms are not grouped together, it is possible to miss important safety signals. Next slide, please. So then a collective way forward would be to develop standardized set of queries that we can apply, and in order to determine what queries or what medical concept to start with, we decided the first approach would be to see what are the most frequently encountered terms in the labels. To find out that, we use natural language processing, and we processed more than 38,000 labels of 1,254 active moieties and took the top 60 terms to start with. Of those 60 terms, we ended up developing queries on 54 of them. There were a few terms that were not included, such as death, it's very absolute, pain, which is very non-specific, and it can be very general. But for the rest of the 54, the first phase of FMQ development developed the queries on them. Then we went to our review divisions and asked, are there any medical concept that they are dealing with on a regular basis that they would like us to develop queries on? So we received quite a few recommendations from the divisions to develop queries on. And we also evaluated existing query systems. So we have the standardized MEDRA queries. Later on, you'll hear from Dr. Ellis Unger about his set of queries. So we've evaluated those. There are other queries that are available, DMEs, MLGs, there are other queries. And we felt that for pre-market setting, none of these existing queries really met the need, and therefore we established the FMQ working group and collaborated with 80 reviewers across divisions. And this really is a great strength of this FMQ medical queries or FDA medical queries, because these are the reviewers who are subject matter experts. So I'm really happy to say that we launched 104 FDA medical queries. Please note that 4 of them are algorithmic queries, and that is really a step forward, as algorithmic queries not only look at data from adverse event analysis, it also combines data from lab, the concomitant medications, the medical history, and temporal association. However, this is a recent development, and so these 4 algorithmic queries are developed but have not been launched. Rest of the queries have been used, and we have also incorporated feedback from the reviewers in the FMQs. And we really look forward to getting some feedback from all of you so that we can make it even more refined. And once we developed those queries and started using it, there was a need for a set of recommendations for use of this in labeling. So with the help of our Medical Policy Council, we do have the recommendations that Dr. Eric Brodsky will talk about later in this presentation. Next slide, please. So the importance of grouping similar preferred terms is not a new concept. It was already published in 2005, March, in the Guidance for Industry in the Pre-Marketing Risk Assessment. Next slide, please. So the FMQs are standardized groupings of these related preferred terms, which are developed by the review staff. MEDRA PTs are highly granular with more than 24,000 PTs. So FMQs actually focus more on medical concept. So for example, initial insomnia, middle insomnia, early morning awakening, they all are basically a different way of saying that it's insomnia. So they're all combined for the medical concept of insomnia. So the goal is to improve safety signal detection in clinical trial data sets with a standardized approach, which will hopefully increase the efficiency and consistency. Next slide, please. So let's take a look at the example of how this is helpful. So if you look at the left side graph, it shows the analysis of single PT for anxiety, and the right side graph combines panic disorder, panic attack, nervousness, generalized anxiety disorder along with anxiety, because that is part, all of these are related to anxiety. As you know that we cannot really include all the adverse events into the label. So in order to determine, there has to be some cutoff. And different review divisions, depending on the need of the NDA, determine different cutoff. So in this case, 2% cutoff was decided. And if you look at the left-sided graph, just looking at a single PT does not make that cutoff. But on the right-sided graph, when you combine these related PTs, it does make a cutoff, and it will be, in this case, it will be included in the label. So that's one of the implications of looking at the group PTs. Next slide, please. Okay, so the FMQs are designed into narrow, broad, and algorithmic queries. And so let's look at the concept as a high level. So the narrow queries are very specific for the medical concept. So indicates that the FMQ occurred with more than 90% probability. And we'll look at the ground rules related to this. The broad FMQ terms cast a wider net. And so they are less specific, but they do provide reasonable assurance. So maybe 30 to 50% probability that the medical concept occurred. And as we try to group these preferred terms, in fact, broad terms were a little bit of more discussions and more difficult than the narrow terms. Narrow terms are usually, people do have a good idea that it is very similar to the actual concept. Here, the algorithmic queries are an important next step, and it uses data from multiple different data sets. I won't go into the detail, as Dr. Scott Prostel is going to talk more about this later in the presentation. Next slide, please. So now let's look at the ground rules. We needed to design ground rules because we are applying medical judgment to the preferred terms to group them under a medical concept. In order to do this consistently, there was a need for ground rules. So here are the ground rules for the narrow queries. So the PTs that are near synonym of the FMQ concept, for example, abdominal discomfort in FMQ abdominal pain, is considered a narrow PT. So PT that are subgroup, meaning anemia neonatal, is also, it's just a subgroup of anemia. So it is included as narrow. The PTs that specify an etiology for the FMQ concept, such as uremic pruritus, is, a pruritus. And so therefore it is included as narrow in FMQ pruritus. And then the PTs that ensure the occurrence of the FMQ concept. So we couldn't think of the situation where there would be an aortic rupture, but would not be a hemorrhage. In most cases, in more than 90% certainty, we can say that if aortic rupture occur, that means hemorrhage occurs. So that's why it is included in the narrow queries. Next slide, please. Now, the broad queries are reasonably suggestive. So for example, PTs that may result in FMQ concept like osteopenia in the FMQ osteoporosis, but that's osteopenia is not the same or similar as osteoporosis. So therefore it's not narrow, it's broad. PTs that provide lab, radiologic, or other diagnostic test results that are reasonably suggestive of an FMQ. So for example, blood glucose abnormal. It is included as broad in hyperglycemia and also hypoglycemia because we don't know whether abnormal means increased or decreased. The PTs that are reasonably suggestive of FMQ concept but not required by the FMQ concept, such as bronchospasm in the hypersensitivity, are also included as broad queries. In the PTs that indicate a carrier status, such as bacterial disease carrier in the FMQ bacterial infection, are also broad queries. Next slide, please. Now, there are some PTs in MEDRA that are just too vague, so they are excluded from the FMQs. So for example, PTs that are neither a required component or reasonably specific. And in this case, actually, this is more related to FMQ concept. So nausea would not be included in migraine because it's not specific nor a required component. But what I meant by PTs that are too vague is here. So some of the PTs are like this, the PT Clostridium test. So we don't know whether it was positive or negative. And if that is produced without a result, then we would exclude it. However, when the PTs provide test name without a result, but would only be performed in the presence of disease, it should be included if they otherwise qualify. So for example, if there is antipsychotic drug level reported as a PT, then it is included in the as FMQ psychosis as a broad because they would not have checked the antipsychotic drug level if the patient did not have psychosis. We also note that antipsychotic drugs can be used in other conditions too. Therefore, it is included in broad. But the fact that they check the drug level does mean that there is a reasonable probability, and that's why it is included. Next slide, please. So now let's look at how the FMQs were constructed. So the FDA review staff, who are the subject matter experts, the therapeutic area experts, developed the standard groupings of related AEs. Each FMQ represents a distinct medical concept and they stand on their own, meaning the FMQ for nausea and vomiting will have quite a few PTs that would be overlapping, but we decided to have each FMQ stand on its own. So if any PT that relates to nausea, they would be included in nausea and as a narrow, but then for the vomiting, it may be included as a broad for the vomiting. Now, each preferred term was independently adjudicated by a subject matter expert reviewer. So any discrepancies were adjudicated by the working group. So each term had at least 2 people independently adjudicate them. And as I described earlier, the ground rules were created to apply the medical judgment consistently. And still there were some difference of opinions. And so the steering committee made the final decisions. Steering committee also ensured version control, systems development for the FMQs and its connection to different review tools we have. Also, we do up versioning with each MEDRA release at the major release. So we do, with each up versioning, set of new PTs are released, and we um, go back and align them to all the existing FMQs that are already developed. The change control is also very important, which is also overseen by the steering committee. We took a cumulative approach. So we included current PTs, former PTs, misspelled terms that were reported to us in the clinical trial data set. The rationale is that, even though, let's say, an FMQ may have 100 preferred terms, but the data set that's submitted as part of the new drug application, the AE data set may have maybe just 20 terms that matches that FMQs. So it will pick up what's there, but if it is not there, then we don't really have to worry about that getting into FMQs. So we have taken the cumulative approach in the formation of FMQs. Next slide, please. So then there could be a natural question, because we do have SMQs, what is the difference between the FMQs and SMQs? So FMQs are designed with pre-market safety evaluation in mind. SMQs are more broader use. They're also used in the post-market setting. So for example, FMQ pancreatitis includes alcoholic pancreatitis, autoimmune pancreatitis, obstructive pancreatitis, and viral pancreatitis. The rationale here is that we are still learning about the safety of the drug. And what if drug makes patients more susceptible to alcoholic pancreatitis? So even though the alcohol consumption level may be very similar between the drug group and the placebo, but now we are seeing more patients with alcoholic pancreatitis in the drug group. So therefore, any kind of pancreatitis is included in FMQ pancreatitis. However, with the SMQ pancreatitis, if there is already a cause, then that may be excluded, because in post-market setting, we do receive millions of records. There are some other differences as well. One of the things I can just point out very quickly is on the right side box, is that there are FMQs for which there are no SMQs. So there are 40 plus FMQs for which there are no SMQs. And so in those cases also, it differs. Next slide, please. So here are the 104 FMQs that are developed. I have linked to the docket here. You can get to the detailed document for each preferred terms within all these FMQs at that detailed document. We are also hoping to get your feedback. Thank you. Next slide, please. So now I'd like to invite Dr. Scott Prostel to talk to us about the exciting topic of algorithmic FMQs. Scott.
Hi, thank you, Vaishali, and good afternoon. It's our great pleasure to show to you and go into more detail about the algorithmic FMQs that we have been working on. Next slide, please. So as Vaishali has already told you, our standard FMQs have two components, the narrow and broad, and that applies to all 104 FMQs. But right now, I'd like to go into a bit more depth into the algorithmic component. And that is for 4 of the FMQs, and I'll be describing those momentarily. We're excited about the algorithmic FMQs because they make more and better use of the data that is submitted to us in marketing applications using multiple data sets in addition to obviously the adverse event data. This includes laboratory data, concomitant medications, medical history, and timing or temporal relationships between events. And before going into the development, I wanted to just show you an example of a mock algorithm so that you have more of a concrete image of what these look like. So you can see in the blue box multiple criteria that are examples of what we've used in the algorithms. And so each algorithm really consists of a series of criteria. In this mock algorithm, there's 4. It can be combinations of adverse events. So it might be, some adverse events are in relationship to the FMQ, they're just too broad, they're too non-specific, and so they don't make it into the standard FMQ at all, but what if it happened in a combination with another adverse event and really improving the specificity? So you can see here in the first criterion, really it's a combination of PTs that together are sufficient or that we believe is sufficient for detecting a specific adverse event. The second criterion is just an example of how we might use a laboratory result, perhaps greater than the upper limit of normal, to detect an adverse event. In addition, we could have a PT or adverse event that's in the setting of a concomitant medication that started within 3 days. And that's an example of using timing information. And the last example would be a PT in the setting of a particular medical history, such as diabetes, and that combination being sufficient. So these are just examples, but I wanted you to see the overall structure before going into the development. Next slide, please. So this was our development process for the algorithms. And we started, if you look to the far left, the initial development of a proposed algorithm, really. That was based on expert opinion and use of the medical literature, which was then programmed so that we could apply it to a clinical trial database of over 10,000 studies. And we would apply the results or apply the algorithms and then look at the results and adjust the algorithm based on our findings in an iterative process. As you can see, it goes in a circle in the middle of the slide. We did use the trial database in multiple ways. Sometimes we used a large random selection of the trials. At other times, we used targeted trials with known FMQ associations, as well as trials with a high prevalence of FMQ terms. And when we were looking at the results, sometimes it was assessing just the total number of patients and types of signals being identified and determining whether or not that was reasonable. At other times, we had access to narrative reports, which we assessed in combination with what we were seeing in the algorithm to see whether or not there was a match and whether or not the results were appropriate. So this was part of our development process. This is not a validation process, but was how we created these first four. We are in the process of validating the algorithms as well, but that is well underway, but not complete. Once we, based on clinical expert feedback, we arrived at what we have now as our final versions, which could certainly change over time. We do see this as a learning process, and we actually look very much forward to getting feedback from you and learning from our experience with applying these to the data sets we receive. Next slide, please. So this is the first algorithm. It's probably our simplest and involves rhabdomyolysis, and it follows the structure as I was talking about earlier. It's a patient qualifies if they meet any of the individual listed criteria. And for all of our algorithms, the first one is any FMQ narrow term for that algorithm. We want the overall algorithm result to be as sensitive and as specific as possible. And so we certainly have a high degree of confidence in the narrow terms, and that's why we include them in the algorithm. The next term uses a laboratory result. So that's an example of using urine myoglobin that's detectable. That would be evidence of rhabdo. And the following criterion is use of creatine phosphokinase, which probably no surprise. There are multiple thresholds in the literature that are quoted. We chose 5 times the upper limit of normal. That's certainly, it's one of the more common thresholds, but certainly not the only. And the last criterion is what's known as the rhabdomyolysis triad. So we certainly thought we should have that. It involves a combination of adverse events, myalgia, muscle weakness, and change in urine color. And we selected 7 days within each other. These all had to occur within 7 days. Next slide, please. So this is a little more complicated, the hypoglycemia FMQ. Again, it follows the similar structure that you saw for rhabdo. And we have as our first criterion any narrow term for hypoglycemia. Again, for the second, we see a laboratory finding. And for the third, it's a combination of laboratory data plus an adverse event term and timing information. So these are events that have to occur within 1 week of each other. And we also have supplemental terms. If you can see at the bottom of the slide, the double asterisk, you can see a number of terms that we wouldn't have been able to make use of but for an algorithmic strategy, because these are simply, if you think in terms of hypoglycemia and the terms that you can see as supplemental, such as accident, anxiety, asthenia, these terms certainly are consistent with hypoglycemia, but they're inadequately specific. So we would not be able to use these terms, but for combining with other information to improve the specificity. Next slide, please. And now we're getting even a little more complicated, or certainly longer. This is the hyperglycemia algorithmic FMQ. Again, the same pattern, a series of criteria. If a patient meets any one of them, they are considered to have met the standard for the algorithm. The first is any narrow term from the hyperglycemia FMQ. And then you see a series of laboratory criteria. And if you look at criterion number 4, that uses concomitant medication. So that's the first time we're doing that. And we're using the indication and class data sets for the concomitant medications. And you can see terms that would include you in the algorithm and terms that would exclude you. And you can see a lot of these are not complete words. This is because there was a fair amount of creative spelling in the data sets, and we needed to maximize our sensitivity and specificity. So we're really using search strings for some of these to detect what we're looking for and not miss any information. The remainder of the criteria you can see are again, laboratory information, obviously related to blood glucose and hemoglobin A1C to detect these hyperglycemic events. Next slide, please. And the final algorithmic FMQ, which was I would have to say, a Herculean effort on the part of the working group. They were able to take a very complicated type of adverse event and simplify it into what you see here. So this involves really 4 columns of adverse events that are much longer than are displayed here. I just truncated it so that I could put it on a slide. But category A are the narrow PTs. Those are the same preferred terms or types of preferred terms as for any other FMQ. And then categories B, C, and D represent adverse events consistent with hypersensitivity for different organ systems. So category B is respiratory, category C involves the skin, and category D are systemic reactions. So if you look at the top of the slide, again, a patient qualifies based on any of these 4 criteria. Category A, again, is a narrow preferred term, so we include it. And otherwise, a patient qualifies with based on combination or any combination of category B, C, or D. And the events have to have a start date within 7 days of each other to be included. Next slide, please. And that's all I had to say about the algorithmic FMQs. A lot of people have worked on these, and we're certainly very appreciative. There are too many really to name individually, but you can see here the core working group members listed and the steering committee. And on the next slide, you can see additional people who have contributed to the development of these. And again, we're very appreciative. Thank you very much. Next slide. So Dr. Brodsky will be sharing with you a strategy we've developed for how FMQs would be labeled. He's Associate Director of the Labeling Policy Team within the Office of New Drugs at FDA. And so Eric, please take it away.
Thanks, Scott. Good afternoon to everybody. So as Scott stated, we are going to, the discussion this morning was about FMQs, development of FMQs, safety analyses. We're going to turn our attention to how to include group term information as well as component term information in the prescribing information, specifically including it in the common adverse reaction table in the clinical trials experience subsection of section 6, the adverse reaction section. Next slide, please. And here's my disclaimer. So in addition to talking about how to incorporate group term information in labeling, such as FMQ and their component term information in labeling, we'll update you on new prescription drug labeling resources for industry, as well as healthcare practitioners and patients. Next slide, please. So this is for the purposes of prescription drug labeling. There are different definitions of adverse events and adverse reactions, but these are according to our regulations and guidance. So according to our guidance, an adverse event for the prescription, for the purposes of including information in prescription drug labeling, untoward events that associated with use of the drug, irrespective of causality. Adverse reactions are adverse events with a possible causal relationship to the drug. That is according to our regulations for the physician labeling rule. I should note that our old format labeling regulations from 1979, the adverse reaction definition is more closely aligned to adverse events. So when one PLR converts or changes the labeling from old format to PLR format, there's a different definition of adverse reactions. Next slide, please. So this is well known to many folks. This is basically factors to determine whether an adverse event is an adverse reaction and is appropriate to include in labeling. And this is stated very nicely in the adverse reactions section of labeling guidance. Next slide, please. So as I discussed before, the talk really involves the most common adverse reaction table or tables in the clinical trials experience subsection of the adverse reaction section, section 6, in the full prescribing information. Section 6 is one of up to 18 sections in the full prescribing information. I should say for the purposes of this presentation, I'll be mostly referring to the common adverse reaction table, but as you know, sometimes it's appropriate to have several adverse reaction tables, particularly if pooling is inappropriate, potentially different durations of trials, different dosages, different populations, adult versus pediatrics. Next slide, please. So this is a fictitious example of a common adverse reaction table that is included in subsection 6.1, clinical trials experience subsection in the adverse reaction section. I should note that it is required in the clinical trials experience subsection to identify adverse reactions that occurred above a specific rate. And according to our guidance, we say that it's reasonable to include this in a table for improved presentation. So let me orient this table to you. So as you know, we have the table title, obviously very important to include what it's including, the patient population, the duration, the study. Sometimes when I review labeling, I add these items. So really important to when you're developing labeling to think about all of these elements. The items in blue are group terms. So they could be any type of group terms. The items in pink are preferred terms that are determined to be adverse reactions that are part of the group term. So the group term appears in the listing in this fictitious example in blue, and the component terms that are determined to be adverse reactions are listed in pink. The green terms are individual terms that are not grouped. So as you noticed in the listing, you may have group terms as well as individual terms that are not grouped. Next slide, please. So we talked a lot about, Dr. Stein, Rochelle and Scott talked about the benefits of grouping terms. I'll just mention one more item. The last item here is that sometimes we group terms. Sometimes we think we see a signal, but then we group terms and we determine it's not a signal. That's also an important feature. So potentially without grouping, you think it looks like an adverse event, but after grouping, you determine it's not actually an adverse reaction and you don't include it in labeling. Next slide, please. So the adverse reaction section of labeling guidance also talks about grouping. It talks about if an adverse reaction represents the same phenomenon, you should group it as a single adverse reaction. So this would be an example of sedation, somnolence. Some of the concepts we talked about so far during this discussion. Additionally, the adverse reaction section of labeling guidance talks about if you report adverse reactions in more than one body system, but it's part of the same pathway physiology, it may be more appropriate to characterize that as one adverse reaction. So if you have a patient that has acute nausea and vomiting, their amylase goes up 3 times greater than normal, they have epigastric pain, and maybe their lipase goes up, it may be more ideal to name the term acute pancreatitis rather than the individual terms. Next slide, please. So in the next series of slides, I'm going to talk about 4 fictitious labeling examples. These examples are mostly based on if you have placebo-controlled studies. I recognize that not all studies are like that. We frequently have single-arm studies. We have some active control trials, and there are some additional considerations for those designs. After we discuss these labeling examples, we'll summarize the principles of this labeling paradigm about how to and when to include group term information as well as component term information in the adverse reaction section of labeling. Next slide, please. So this is great. I'll first orient you to these next series of slides. This first box here represents your analysis. This is not in labeling. And again, this is a fictitious analysis. So for the purposes of this, we're taking the FMQ anxiety group term. Here you see a potential signal, 7% versus 3%. The green terms are some of the component terms. These component terms are near synonyms of the group term anxiety. And you could click through. So this part, thank you, this part right here, the right part the right table is what would go in labeling. So this would be the most common adverse reaction table in subsection 6.1, the clinical trials experience subsection in section 6. And then here you would do, you could see anxiety is 7% versus 3%. Now, if you look at the green terms, they're not specifically named. However, they are incorporated into the incidence. 7% and 3% versus 3% represents the component terms. Note that we do include a footnote. Anxiety, you could say footnote number 2, and we identify to healthcare practitioners that they're composed of several similar terms, because that's slightly different than vomiting, diarrhea, dermatitis. Those are not group terms. And next, you could click through. So here are the principles that I just stated. Next example, next slide, please. So in examples 2 and 3, we're going to talk about situations where we include information specifically in labeling from the group term as well as component terms. The difference between 2 and 3 is that 3 are more consequential or adverse reactions of more clinical importance. So this example here, again, on the left side, this is not in the labeling, this is one's analysis. The green represents the group term, and the terms below are the component terms. And so you see a differential between the drug-treated patients and placebo-treated patients in the group term overall, as well as 2 terms, social phobia and stress, that will meet the criteria for inclusion in the adverse reaction table. And you would click through. So this right here, again, represents what would appear in the labeling. So here we include the group term anxiety, and all the component terms are represented in that incidence 12%. Specifically in the footnote, we identify social phobia and stress because they meet the definition of an adverse reaction. And here in the footnote, we include the definition, the criteria, I should say, of how we determine inclusion of the adverse reaction in this table. And you could click through. So the principles, as I stated before, the anxiety group term is included. The social phobia and stress are included not only in the overall incidence 12%, but they're named in the footnotes. Next slide. So number 3, there's 3.1 and 3.2. These represent a similar situation where you're going to include information about the group term as well as the component terms, naming them specifically in labeling. The difference between 3 and 2 is 3 are more clinically important terms. So panic disorder, obsessive compulsive disorder could be considered more clinically important than the other terms that we used before, such as stress. 3.1 versus 3.2 presents 2 different approaches to how to identify these clinically important terms in the body and or footnotes in the common adverse reaction table. And you could click through here. So again, the group term here, this is signal 9% versus 2%. It's included within the body of the common adverse reaction table. These important terms that are not near synonyms, panic disorder and obsessive compulsive disorder, approach number 1, 3.1, are included in the footnotes. Note that for this fictitious example, I'm trying to demonstrate a very lengthy adverse reaction table. So potentially one would less likely want to include those clinically important terms within the body just because of the length of the table. And you could click through here. Again, the anxiety group term is mentioned, and we include the clinically important terms in footnotes. Next slide, please. So 3.2 is the same situation, same data. 9% versus 2%. We've identified two terms that that basically are important, clinically important. And here we're going to include those terms within the body of the most common adverse reaction table. And you could click through. So right here, we include those terms within the body under the anxiety term. Note that we include a footnote, and we basically acknowledge the panic disorder and obsessive compulsive disorder, but that group term includes other related terms. And for this fictitious example, I purposely made it very short. This would be a situation that you could consider including these clinically important terms within the table. And you could click through. And we've talked about these principles. Next slide. Next slide. So this slide presents fictitious example number 4. And this is a situation where the signal is only coming from one or more component terms. And actually, there is no, if you wipe away panic disorder and obsessive compulsive disorder, you don't see a signal. Again, this is a fictitious example, and you could click through. In this scenario, you would not include the group term because the adverse reactions are panic disorder and obsessive compulsive disorder. And that's what we would include in the labeling. Again, all these principles are based upon the adverse reaction section of labeling must include adverse reactions. We don't include adverse events that are not adverse reactions. And you could click through. And I discussed those principles. And next slide. So here is a summary of the paradigm. So as I stated previously, very important, the whole concept are including group terms and potentially component terms that meet the standard of an adverse reaction. As exemplified in the prior example, example number 4, if the group term and component terms meet the definition of an adverse reaction, but the signal is only driven by the component term or terms, one only includes those component terms in the most common adverse reaction table. Next slide. Next principle. So as I stated earlier, if we're including a group term, those component terms are represented. They're represented because they're components of the incidence. Additionally, you could click through if the component terms are near synonyms, and I think that was the first example. They're not specifically mentioned in the body of the footnotes. For example, like anxiety, NES, or other synonyms, they're not specifically named. In contrast, if you look at those were examples 2 and 3.1 and 3.2, if they're distinct clinically events and they're not synonyms, then they would be included in the common adverse reaction table, and we discussed considerations about whether they would be included in footnotes or potentially in the body of the table. Next slide. Now, actually, we're going to turn our attention to as I discussed, we have new prescription drug labeling resources, and the resources are directed to industry primarily, but we also have resources for healthcare practitioners and patients. Next slide. So this is our homepage. This includes labeling databases such as FDA Label, Daily Med, Drugs at FDA, and searchable product databases such as biosimilar product databases, Orange Book, Purple Book. Additionally to this homepage, we have a specific page on the prescribing information that obviously includes safety-related information in the prescribing information, specifically patient labeling, carton container labeling, generic drug labeling, biological product labeling. Next slide, please. I'm going to show you the specific page for prescribing information. We designed this page to follow the required sections of the full prescribing information. You see the highlights of prescribing information, and then it follows. So all of our guidances relevant to each of these sections are included. All of our public presentations that are relevant, we've included that here. Next page. So here you could, if you're interested in adverse reactions, as soon as this is over, we'll also include this labeling presentation under presentations. That's our adverse reaction section of labeling guidance. We published in February the immunogenicity labeling guidance. That is here, as you know. The recommendations are to include immunogenicity data in subsection 12.6 rather than section 6. And so you could see our recommendations in the immunogenicity labeling guidance. Next slide. Turning our attention, this is our new webpage for healthcare practitioners and patients, written in, I would say, more patient-friendly terms. Thank you. And I'm going to turn it back over to Maureen. Thank you.